Hi everyone. In this video, I'm going to teach you the technique on how to do a smear and a simple stain. When I mean a smear, I just mean how do I get a sample, some kind of sample of microbes or an environmental sample on a slide and dried down onto the slide. And then at that point, the cells don't have a lot of color. So frequently we want to add some color with some kind of stain. And the technique of a simple stain will just stain all the cells the same color. So it's a really basic microbiology technique that everyone should know. Everyone that works in a microbiology lab should know. So in this one, we're just going to take some cells from our own bodies and we're going to do cheek cells. In the lab that I have set up, it says to do a simple stain of cheek cells and of some yogurt. So we do both of these smears and stains in a very similar way. So I'm just going to show you the cheek cell smear. And when you do this in lab, you'll put some cheek cells on one side of your slide and you'll put a really thin layer of yogurt on the other side of your slide. So get your slide. If it looks dirty at all, you can grab some lens paper and wipe it off. And I'm going to get a sterile stick. So you'll have some sterile sticks available to you. Kind of shake one out. Try not to touch the other ones if you can help it. And put the lid right back on so the others remain sterile. And I'm going to get some cheek cells. And they're going to be from inside of my cheek. You can go around your teeth, on your tongue even. But something like this. Like that. And it wasn't very invasive. I didn't have to scrape very hard. And I'm going to get my slide and I'm just going to rub that stick right on my slide. And even with that really tiny bit of rubbing that I did, I can actually, you can see it in the video. Let's see. If I tilt the slide just right. You can see that there's some kind of cloudiness on the video, on the slide. And those are my cheek cells. Okay. On the cheek cells, there will be bacterial cells. So hopefully you'll see eukaryotic cheek cells and prokaryotic bacterial cells. So I'm going to let that dry. When you do this experiment, you're also going to go to the other side of the slide and you'll get another stick and you'll put a really thin layer of yogurt. Let both of those dry. Make sure you remember which side is up. When I'm done with my stick, I'm going to put it in a disposal beaker just to get rid of it. Once these are completely dry, if I add a stain to it, the liquid might rinse it off. So we do something called heat fixing, which gives me an extra dryness to my cells and helps them stick to the slide. So we always want to heat fix our sample before we stain it to help the cells stay stuck to the slide. Now I'm just going to set my slide on the edge of the beaker there, and I'm going to turn on my Bunsen burner, which we learned how to do in a previous video. There we go. And to heat fix, all I'm going to do is I'm going to hold my slide at one end, and I'm going to go to my flame, and I'm going to pass my slide through there once, twice, 3 times. So I warmed up the underside of my smear. And that just gives it a little bit more drying that will really help the cells stick to the slide. Make sure it's cool enough, and you can switch your hand to the other side, and you can heat fix the other smear you made. 1, 2, 3. Okay. When I'm done with that, I turn off my Bunsen burner. Okay. So now you have your cheek cells and your yogurt heat fixed to your slide. And what I'm going to do is take a stain. In this case, we're going to use a stain called methylene blue. There's a lot of different stains you can use to do a simple stain. Methylene blue is one of the common ones. And I'm going to make a puddle of methylene blue on each of my smears. And make sure you cover all the cells and let it sit there maybe about 30 seconds. The time isn't really critical, but 30 seconds seems to do the trick. Now you might want to hold your slide. In fact, I highly recommend that you hold your slide with a clothespin. You'll notice my clothespins are all purpley and blue looking. If you do it without a clothespin, your fingers might end up looking like that. So after about 30 seconds, I'm going to take my slide and I'm just going to dribble the stain into my waste beaker. So I need to get rid of the excess stain. Then carefully get a wash bottle that has deionized water in it. Be careful not to use the Lysol disinfectant bottles. Sometimes the colors in the dye can actually change the dyes in the disinfectant can change the color of your smear, of your stain here. So here I've got a wash bottle, and I do this kind of waterfall technique. I don't want to spray right on my smears because I don't want them to come off, but I do kind of do a waterfall where I start at the top and I just let the water dribble down. And I want to get as much of that stain off as I can. You'll notice the bottom, there's a little collection of stain at the bottom. Sometimes rinsing off the back can help get rid of a little bit of that. And that looks pretty good. But now I've got a wet slide. So Bibulus paper comes in a little booklet like this, and it really just is blotting paper. And you'll notice if you look in there that a lot of the pages already have some colored stains on them. And so you don't need to pull out those pages. Those pages can be reused again. It's really, the booklet is just designed to put my slide in there, lay it in between any two pages, close the book, and then press it. Notice that I took my clothespin off, because if you have your clothespin in there, your slide will be at an angle, and when you press it, the slide will break. So blot the water off your slide. And now I've got a slide that's nice and dry, and I can view that under my microscope. You can view this all the way up to 1000x because you can put oil right on top of those smears. Good luck. This is a technique that you should get pretty good at if you were to become a microbiologist.